My name is Sophie and I work at the University of Birmingham. My job role is postdoctoral research fellow, which means I do my own research project, but it's as part of a larger lab under a professor. My research is into blood cancer, and what we're doing at the moment is trying to work out why sometimes when we give patients medicine, their cancer will go away and they'll get better for a bit, but then it'll come back and it'll come back worse. That's really bad. So in more detail, I research a type of blood cancer called acute myeloid leukemia. And one of the real problems with this disease is that when you treat patients with chemotherapy, the disease will go away in something called into remission, but then it will come back, which is called relapse. And unfortunately, often when that happens, patients don't survive. And what we've found is that there's a type of stem cell which lives in the bone marrow, and this can actually hide from the chemotherapy and cause the cancer to come back. And what we found is that it can hijack the body's own messenger proteins, and it can use this to kickstart the cancer process all over again. So what we're trying to do is actually test drugs to block this process so that we can stop the cancer coming back. A day in the lab for me usually has a lot of different experiments. So one of the main things we grow up the cancer cells so that we actually have enough to do experiments on and to test drugs on. And we do a lot of something called genomics in our lab, which is looking at how genes are expressed by doing something called next generation sequencing. So we look at every gene in the cell. And this is quite complex, so it involves a lot of long experiments with tiny, tiny volumes of liquid. But it also means that we get to do more fun things. So we have a technique which involves pulling out special bits of the DNA that we're interested in, using magnetic beads, so they stick to a magnet in the tube. And that means we can actually look directly at the DNA to see what's going on. And then we do a lot of analysis, so comparing it actually to the genome. So one unexpected and exciting thing that's happened to me through work is actually due to our links with the group who fund my research. So I'm funded by a charity called Blood Cancer UK, and they actually came to visit our lab recently, and they brought someone who'd done a lot of fundraising for us. And he's actually a member of the Leicester Tigers rugby club, and he came for a lab tour and to meet us all and just to see where the money he's raised is going and what we're doing on a day-to-day basis in the lab. That was pretty cool. So my career path into this role, I first did a PhD in stem cell research up in Manchester. While I was doing my degree, I actually worked part-time at a biotech company, and they work on stem cells in various diseases. And this really sparked my interest into stem cells. And I also, during my undergraduate degree, worked on breast cancer stem cells. So I really knew that this was where I wanted to be. And when I finished my PhD, this job was advertised, working on stem cells in cancer. And so I've moved down to Birmingham, and I've actually been here for 6 years now. The advice and reassurance I would give to my teenage self would be that as long as you stay excited about science, it's definitely possible to get anywhere you want in it. But don't get too fixated on working on the Human Genome Project, because they'll have finished it by the time you graduate.